All right, and welcome to episode 26 of the Breaking Bio podcast. Today is April 11th, and I'm Stephen Hamblin, postdoc from the University of New South Wales in Sydney, Australia. And so we've got Morgan with us today.
Hi, I'm Morgan Jackson. I'm a PhD student at the University of Guelph in Ontario, Canada.
And a special guest who has put up with our complete train wreck of a morning here. We have Siouxsie Wiles with us.
Hi, I'm Siouxsie Wiles from the University of Auckland in New Zealand. And I'm a microbiologist and obsessive over things that glow in the dark.
We're here today because you do awesome science. And I had a look at that video that you produced, the Astro Squid.
Right.
So I should do the whole what do you do, do the traditional method here, but I'd like to get right into the Astro Squid.
Sure.
So why don't you tell us a little bit about how that came about?
So the idea for doing some animations came after meeting a guy called Daniel Keogh a couple of years ago now. So he is an Australian science communicator, and he had done this incredible animated video about the placebo effect. And it was just awesome, and it kind of went viral on YouTube, and I thought, wow, I would so love to have a try at that, at basically learning how to tell an interesting story that's, about science in like 3 minutes. And so I contacted his animator, Luke Harris, and sort of said, so, how much do you cost? And he told me, and it's about, I think it was something like 500 Australian dollars per 30 seconds of footage. So I was like, wow, okay, right. So I kind of went around and asked, actually in my institution, whether they would whether they would just let me try this thing to basically make an animation. And originally I wanted to make an animation about fireflies and NASA. Because the thing about kind of glow-in-the-dark organisms is they're really cool, so they all glow in the dark and they all do it for different reasons. But they're also a kind of into cool science because scientists use bioluminescence in so many different ways. So my institution said, yeah, okay, we'll let you have a try, but it's got to be about your research. Now, I work on infectious diseases and tracking bioluminescent bacteria within animals. So I kind of said to them, you do realize what I do, right? And they were like, yeah, that's fine. I said, so we're going to make an animation about using animals in research? And they were like, yeah, that's fine. And I kind of just wondered whether they were either weren't listening or didn't think it would happen. So that was the first lesson they learned about me, is that I tend to do stuff when I say I'm going to do stuff. So they gave me money, and I went away, and with a lot of help from my husband, who's a mathematician, and various friends, kind of put this very short story together. It was like 2.5 minutes that explained how awesome fireflies were, sort of how they glow and why they glow, and then how I use their light in my research into infectious diseases. And it kind of ended up being about tuberculosis and how scary and horrible tuberculosis is. And so that did quite well. And I had been a little bit sneaky, and I had also asked a few other places for some money, some charities and things. And so what ended up happening was I got a little bit more than I needed, so I got Luke to make me the ending that I wanted about NASA, because it turns out that NASA very indirectly use fireflies in the search for extraterrestrial life. So we ended up with these two animations that start with how fireflies glow and why, and then one of them talks about TB, and then the other one talks about NASA. And I just thought they were fab, so I really enjoyed the whole process, and I thought I'd love to make some more. And actually what happened then was somebody at a society in the UK called the Society for Applied Microbiology saw them and said, we have a public engagement fund. Would you like to apply to us to make another one? So I was like, yes, absolutely. And what I thought was, okay, so this is a microbiology society, it's got to be microbiology. So I decided on the squid, which is this Hawaiian bobtail squid. They are awesome, and they have a symbiosis with a bacteria that glows, and they use that to essentially, it's like an invisibility cloak. So they hide their shadow because they're nocturnal. They hide their shadow from any prey or any predators. And then I linked that with the studies of this symbiosis and this this bug, Vibrio fischeri, led to the discovery of how bacteria communicate with each other. So this thing we call quorum sensing. So we then basically made this little animation about squid and quorum sensing, and kind of how scientists hope that quorum sensing will, or if you can attack quorum sensing, you could stop some nasty bugs from causing disease. So in under 3 minutes. And that was kind of really interesting because it's science that I'm really interested in, but it's not, my thing. So with kind of some trepidation, I then sent the animation when it was finished. So it was only finished a couple of weeks ago. I sent it to like the big wigs in the field who work on quorum sensing or who work on the squid symbiosis, with a kind of caveat saying, apologies for any liberties taken to fit the story into 3 minutes. And they were all lovely, and they said, how actually it was incredibly accurate, and that they were very impressed with, that I got it all in. So yeah, so I now have money. I've got to make one more, which is going to be about the New Zealand glowworm, which is also pretty awesome. It's this kind of cannibalistic creature that has orgies and all sorts of things. And I'll probably link that rather tenuously to how we study circadian rhythms. So we can basically hook up the genes for light to promoters that work on the circadian clock, and just sort of explain that. So yeah, and then there's loads more. So then I've got to kind of think about whether I'm going to apply for any more funding to make them. But it's a complete side project, so it's the kind of thing that I do of an evening is write these stories up. But it's awesome. I love it. And with funding being so tough here in New Zealand, this seems to be quite easy money to get. So if they won't fund my research, at least this way I get to tell cool stories about science. So yeah, that's how it kind of came about.
Sorry, I did notice that when I was watching the video, is that the story was actually, it was fully detailed, and there was, like you even had the chemical reactions and things that you might not normally see in a video aimed at the public. And I was really impressed by that, right? That you managed to work it in and make it seamless, but still accessible. And I'm wondering how much of a struggle that was. Like, did you have to fight with it?
Yeah, a little bit. So when I first wrote that script, it was actually a lot more detailed, really about how so in terms of the quorum sensing bit, exactly how the autoinducer works, and on a genetic level, basically it comes in and it binds to something else, and then they bind to this bit of the promoter, and that kind of switches everything on. So I kind of had all this, and it was just like, so the scientist in me has to kind of say, do you know what? Actually, that's kind of surplus to requirements. And what are the bare bones? What really are the important things that we want to get across? That we can show visually, that's not too technical, that there's not too many of them. I was kind of quite insistent that I wanted to keep the reaction, the Lux reaction in there. So that kind of just flashes in, and anybody who knows anything about it will say, oh yes, I know what that is. If not, you could just go, wow, that's quite complicated how they make light, that you can just sort of see it flash by, and that's fine. So it is a really kind of tightrope that you're walking between not wanting to overwhelm people with information, but providing enough that people can see, actually, this isn't really simple. This is actually quite complicated, but it doesn't have to be complicated when you explain it. Does that make sense?
Yeah.
And working with the animator is a really interesting experience because they're not scientists. And in fact, with the squid one, it was hilarious. So I was trying to explain to them, what happens, how this counter illumination works. So, you've got a moon that's shining down, so anything underneath that moon, and I was trying to explain it like a torch, anything underneath it will then cast a shadow. So what the bacteria does is or the squid does, is it has these bacteria that basically then shine light down so that it doesn't cast a shadow. And I was kind of doing all this stuff, and I was saying, maybe kind of visualizing it with torches and things. And the first draft they came back, the squid was holding the torch over the top of itself. And I was like, no, that would make an even bigger shadow. What are you talking about? And so they were like, oh, sorry. And so I kind of tried to explain it again with this torch things. And then the next time, the torch was pointing upwards. And I was like, no, that would still be wrong. There would still be shadows. And finally, on the third time, it was like, turn it round. It points down. And I'm like, right, okay, we get that now. So those, and like with the firefly ones, it was really interesting because I had wanted to be accurate in what they looked like and the color of the light that they produced. And it ended up being that the animator kind of took some liberties with that. And I just had to say, do you know what? Nobody except an expert on fireflies is going to notice that. And hopefully they won't be too offended. It'll be okay, and they'll kind of understand that it's artistic license. But the scientist in me was still kind of cringes a little bit when I see that. It's like, oh, it's not quite right. But nobody else will notice. So, yeah, that's a tricky thing. But and I think as scientists, we really struggle with that kind of everything's got to be perfect. And at some point, I think when you become more of a communicator, or you realize that that's kind of important, and some of these other things you maybe just have to make slight compromises on. But hopefully not too many. I guess that's my aim, is to try and not make too many compromises. Yeah, but it's interesting.
So you do a lot of outreach, like you've got a lot of blogging and stuff like that behind your belt. So how is it different going from the written word essentially to more of a script where you're trying to do moving pictures and stuff like that? Was it a different challenge trying to explain science and communicate science in a visual moving media versus the static writing? Yeah,
because you've not got much space. I mean, that was the thing. It was also, the blogging has been really good for me because I suffer terribly from verbal diarrhea, and I can just write. And also as scientists, we write really badly. I mean, now I cringe, and when I look at scientific papers and I think, God, what the hell are we trying to say? Who understands this shit? So actually the blogging has been fantastic, because now it's like, right, okay, so you're talking to somebody who knows nothing. So what is it that they need to know? And I'm trying to start writing my papers like that. It's going to be hilarious. I'm kind of approaching them as though it was a blog post. So what is the information we would need to use? What language would you put this in there so that a non-expert or an expert in a different field could understand? And it'll be interesting when I submit my first one, whether it just gets a kind of, what the hell is this? This is too popular science kind of thing. Yeah. But yeah, so the blogging has been good to kind of teach me how to stop writing like a scientist. And then you've got to go that whole extra step of, okay, so you've managed to get from like 1,000 words to 100 words. How do you now get that sentence down to 10? And what are the bits that you chuck out? How do you, and also you have to make it engaging, which again, we're just not good. We don't write in an engaging way. And I find it really interesting with the blogging and stuff. I'll be looking on Science Daily or something, and I'll find what looks like a really interesting story. And then I go and read the paper, and I'm like, oh my God, this is really boring. How can they make it sound so boring? So that, there is quite an art to trying to make something really engaging. And I'm just learning all the time the language that you use, how you kind of try and, how you, like this thing, with the squid, the process is called counter-illumination, which is a kind of technical term, and it sort of is what it, says what it does. But invisibility cloak is so much more awesome, So there are kind of ways that you can kind of work it. You've got to try and think what are cultural references that you can use that wouldn't end up being too obscure or date too quickly. It's just a whole different kind of way of thinking, and I love it. And I would really hope that, I never want to give up the science, and I really hope that this side of what I'm doing will really feed into how I think about science and how I communicate the science to other scientists. Doing public talks and stuff is completely changing the way I give presentations to other scientists. And then because I've become like a massive kind of, I love live tweeting talks. I find myself going, oh God, this is really boring, or your slides are really bad. Who are you talking to? You're talking to the one expert in the room, as opposed to the 100 other people who have some interest, but you're losing them all. Yeah. So it's really interesting. I think all these things kind of, they're all swirling around in my head, and I'm trying to make use of them, and also reconcile them all as well. It's kind of interesting. Yeah, I'm a jack of all trades, if you hadn't noticed already.
A lot of us are.
Yeah, I've been on that. I've been on that make your talk suck less soapbox for quite a while, and my lab mates are just sick of hearing me say things like, "Yeah, you got to stop just putting a bunch of text on and reading it to the audience.
Yeah. Yeah, I mean, it's the art of storytelling, and so for me, it's the art of storytelling. Whether you're writing it, whether you're writing a script for the animators then to play with. I mean, I have some ideas around the visualization, but I really leave that to them, and they come up with some really cool stuff that I wouldn't have thought of because that's their job. And frankly, if I'm paying them $500 every 30 seconds, then God damn it, they're going to earn their money because I'm not being paid for any of this stuff. So.
That is actually really cool, and I could talk about it all day, but I'd actually like to move on and discuss your current research right now. So, as you mentioned, the Squid animation and the other animations you're doing as a side project. Can you tell us a little more about your main research focus?
Yeah, so let's see. So we've probably got about 3 or 4 different projects going on in the lab. So one of them, and they've basically all involved glowing bacteria, really. So one of them involves making or testing new antibiotics or new therapies, primarily for tuberculosis. So the bug that causes TB is a really slow-growing one. It's dangerous, but really slow-growing. So it takes 4 to 6 weeks to form colonies on a petri dish, and so this means that if you do any assays looking for whether new antibiotics work or not, that takes weeks and weeks and weeks. So we've basically made some glowing TB, and it just means that we can look for light. So if they're dead, they stop glowing, basically. So instead of plating them out and waiting for waiting for the bugs to grow, we just look for light, and so we can get an answer within a couple of days rather than weeks. So we are working with some fantastic medicinal chemists at Auckland who are making anti-TB drugs. It's amazing, really, being in this country, and 2 of the top 10 candidates going into clinical trials for TB treatment come from New Zealand, come from Auckland. It's kind of scary, really. So these guys are tweaking these drugs all the time, and we're basically now saying, right, give them to us, and we'll tell you within a couple of days whether whatever tweak you've done has made a better or a worse drug. So that's one of our projects. Another project I'm really interested in is how is bacterial evolution, and so most of the studies that are done on microbial evolution, like Lenski's experiments and things, where you take bugs, you have them in flasks, and then you leave them, you kind of subculture them every so many days, and you kind of do that for about 10 years. And so the Lenski experiment's probably even longer than that. And then you look at how the bugs evolve, basically, in these different environments. But the environments are really, really simple. And so a few years ago, there was a call kind of from a big name in the area and a really nice review paper saying it's time we did evolution experiments in more complex environments. And I think what they were thinking of was stuff like soil and things. But I was like, okay, I have a complex environment, which is basically a mouse. So one of the bugs I work on is it's like an equivalent of a food poisoning E. coli. So it basically gets swallowed by a mouse. It causes in the mouse strain that we use, it's a pretty innocuous infection. They basically get a little bit of runny poo, but they then spread this bug all over the cage. And if you put another mouse, an uninfected mouse, inside that cage, they will pick up that infection. And so it was like, oh, here's actually quite a nice environment, which is extremely, complicated, but also we could watch. We could basically just do this process where we have an infected mouse, and then we infect another one, and then we infect another one, and you could so have a look at how evolution happens with the bug and under this kind of circumstance. And the idea here also is that you've got a bug and a host that you can manipulate in all sorts of ways. So we can look at mice that have got differences in their immune systems. We could do all sorts of things. So we're really struggling to get that funded, but we are doing it anyway. So I have this poor PhD student who basically hasn't been on holiday for like 6 months because she can't leave the animals. Once you've started it, you kind of have to carry on. And so that's kind of one of our things, which we actually ended up going and trying to raise some money on the internet for. So that was interesting, and we could talk about that a bit later. And then a lot of our work, what are the other ones? Oh, we're kind of doing stuff looking for phage that kill bacteria, all sorts of things. Like, again, jack of all trades, interested in lots of things, but primarily how bugs cause disease and how we can kill them, really, with using a few different examples, all of which glow in the dark. So there you go.
That's cool. So how did you? I think the making TB glow and not waiting until you have to actually be able to count it is freaking brilliant. That's a really cool idea. How did you guys come up with the idea of let's make tuberculosis glow today like that? And where did the glowing come from? How did you introduce glowing to a non-glowing organism?
So people have been doing this for years, and when I did my PhD many years ago, there were already quite a lot of bugs that had been engineered. So you basically take the genes from things like this bacteria that is the symbiont of the squid. You take those genes and you put them in other bacteria, or genes from fireflies and stuff like that, because bacteria are so easy to manipulate. Or some of them are easy to manipulate, and this had been done for lots of things. So because now they're glowing, you can track them, and so there are these fantastic sensitive cameras that allow us to track where the bacteria are. So so just like if you put a torch underneath your hand, you get light coming out the other side. So basically we have cameras that can detect that light that comes out the other side from inside of animals. So we basically have these glowing bacteria, and they're like behaving like a torch inside an animal, so that without, we just put the animals to sleep. In fact, you don't even have to do that. You can just put an animal in this dark box and then say, where are my bacteria, without having to do anything to the animal, and you can see where they are. So are they in the lungs, or are they in the stomach, or whatever, where they're supposed to be, and are they dead or alive? So again, when you're looking at whether drugs are working or not, one of the first things that has to be done is they have to go through animals before they can go through into humans. And normally what happens is, you give the animals this infection, you give them a drug, and then you either wait for them to get sick or not, or you kill them, plate out their organs, and look for whether the bugs have survived or not. With the bioluminescence, you just put them in the camera and say, where are the bugs? Are they there? Has the drug worked or not? And you can use those same animals. You can image them every day and say, right, well, one day after they've been treated, where are the bugs? 2 days, instead of having to kill them, kill animals at each time point. So this technology started in the kind of the early 90s, I think. And when so that was when I started university. And when I finished my PhD in about 2000, it was like, oh, it'll all have been done by now. And of course it wasn't. And there were loads of bugs that hadn't been engineered. And so we have kind of done some of those. And TB ended up being one of the most difficult that lots of people had tried to engineer it and had never worked. And then we got money from the Gates Foundation to basically try this with as many different of these reporter genes that we could, to see which one worked. And I had this amazing postdoc called Nuria who did all this work. She was brilliant, and she got it to work, and now we have these beautiful glowing TB that are really useful, and we can see them inside of animals and all sorts. And now lots of people are interested in using them for both their research and for drug discovery. So yeah, it's cool.
Is there a minimum? Sorry, is there a minimum density for the TB to show up on the camera? Like, is there?
Yeah. So every bug is different because it depends on copy number of the gene, on the promoters that you use, so basically, and what reporter you've used, and then when you get them inside of animals, whereabouts in the body they are. So with TB, it's about, I think it's about 1,000 to 10,000 bacteria that we can see in a spot. But I have other bacteria that it's like 100. So it all depends on what they are and where they are. Yeah, so it's pretty cool. That's with the TB stuff, that's the most sensitive that's ever been reported. So that's good. Although we did say to the Gates Foundation, we'd be able to see like 10, but.
So I'm gonna steal a question from Steve. I don't know if Steven's got another follow-up question or not. But so do you see any? Maybe shouldn't say this, but how soon until this starts showing up in the tattoo enthusiast crowd with, getting glowy bits, essentially, glow bands put under people's skin and thus them glowing? Would it ever be visible to the eye?
I think you can do this already with fluorescence. I'm pretty sure there are fluorescent dyes that you can tattoo in. I mean, it's not going to work.
With bacteria, though.
Yeah. So, well, so the point is the bacteria have to be alive, and they have to be healthy, and they have to be expressing all these genes. And so, that's why they're really useful for us. And it turns out, when bacteria kind of go into stasis and all this kind of stuff, then they either stop producing the genes or they don't have all the cofactors they need. So I get contacted all the time by people saying, oh, I want to make some glowing furniture. I want to kind of make chairs and put all these glowing bugs in it. And it's like, well, that's fine, but it'll probably only glow for about a day, and then it'll sort of stop. And I mean, so the squid solved this problem, right? By every day when the squid, so as daybreak comes, when the squid has finished all its hunting, it expels 99.9% of the bacteria into the seawater. And that way, then it buries itself in the sand to sleep for the day. And that way, during the day, the bacteria then start with their little population, and they now have to double and double and double and double, and until they get to this population density where now, there's enough of them, they've got nutrients and space, and then they turn on their light. Whereas if you just left them, they get to the other side of that curve, and then they go, oh, now we've run out of nutrients and we're dying, and we stop glowing. So in order to make it work for furniture or something, we have to come up with a way of keeping the bugs happy, have space, have nutrients. And I have loads of them, seriously loads of them have said, oh, could we do this and could we do that, and could we pump methane in there? And I'm like, I have no idea. Probably not. I don't know, do you want exploding furniture? I'm not sure.
We're actually come to the end of our allotted time here, and
that was good fun.
We can keep chatting for the after show for a little bit, though. And those that are watching live, which I don't really know if anybody is, they could take advantage. But it's incentive for anybody listening out there to watch live.
we had people popping in during the podcast.
Great.
But no, I want to thank you for spending some time with us today, Siouxsie, especially.
Oh, it's been great.
Given that we had to push things back an hour, we appreciate your patience and your participation. But so, if people want to know more about your stuff, where should they go?
Oh gosh, if you can spell my name and type it into Google, you'll find either me or you'll find Siouxsie from Siouxsie and the Banshees. I'm on Twitter, I'm at Siouxsie W, so SIOUXSIEW. The animations can be found just by going onto YouTube and looking for Skeptimoo, so SKEPTIMOO. We're busy working on a proper lab website at the moment, so at some point that will appear with actual stuff that we're doing and things. And my aim is to try and, when we write papers, to have, actually we're trying to do some of our stuff open, our lab books open, so some of these projects will appear for people to criticize at will and help us keep on the right track, instead of in 3 years' time the reviewers going, oh, you should have done like this. And we're like, oh, we don't have money now. Anyway, yeah. So if you Google me, and I'm all over the place with all sorts of silly things. My blog is called Infectious Thoughts on sciblogs.co.nz, and I try and do a kind of every Monday write about something interesting in microbiology. Although I failed for the last couple of weeks, but.
Well, yeah. There's so much more we could talk about, and I hope maybe we can have you on again to talk about crowdfunding and open research, which has become a bit of a theme on this podcast, actually.
Right.
But for now, if you want to know more about the podcast, you can go to breakingbio.com. And again, we'd like to thank Siouxsie, and we'll see you guys next time.
Bye.
Bye. See ya.